Hello everyone, my name is Gabriela Ruphuy and I am a researcher at UCT Prague. My talk today is about the use of yeast glucan particles as drug carriers. We often relate the word yeast to food, and this is not surprising considering that yeast is one of the most common ingredients used in the food industry. We use it to produce, for example, bread and beer. It is not so common, however, to think about yeast for applications in the pharmaceutical industry, yet this food ingredient has great potential as a drug carrier. In the microscale, yeast looks like the image shown here. They are single-celled fungi, and if we subject the cells to a series of extraction steps, we are able to remove all the internal organelles, leaving only the yeast cell wall. After the extraction process, the yeast cell wall looks like this. It is mainly composed of polysaccharides, particularly beta-glucans, and that is why they are commonly called yeast glucan particles. They are hollow and porous microparticles, they are amorphous, and they exhibit immunomodulatory properties. These properties make them suitable drug carriers. So, there are two possible mechanisms how glucan particles can act as drug carriers. First is macrophage-targeted delivery. This mechanism has been widely studied, and it is based on the fact that because glucan particles are obtained from microorganisms, our immune system detects them as intruders, promoting macrophage phagocytosis. Through this mechanism, drugs could potentially be delivered to the lymphatic system. The other possibility is drug amorphization. Our hypothesis was that given the fact that glucan particles are amorphous polysaccharides, they can improve the aqueous solubility of crystalline drugs via amorphization. So, I am going to focus on this mechanism, and for that I will refer to the solubility problem. Oral drugs must go a long way before they can actually reach the bloodstream. They go from the mouth to the esophagus into the stomach to finally reach the small intestine, where most of the absorption takes place. However, the drug must be dissolved in the aqueous gastrointestinal fluids before they can be absorbed. Therefore, the low solubility of a drug in water leads to a low bioavailability. This is the reason why solubility is one of the main challenges in the pharmaceutical industry. In fact, about 90% of the drugs in the discovery pipeline and over 40% with market approval are poorly soluble in water. One way in which the pharmaceutical companies have managed to overcome the poor solubility of drugs is by producing the so-called amorphous solid dispersions. Because the amorphous state of a solid has a higher free energy than its crystalline counterpart, its solubility is also higher. However, the amorphous state is also thermodynamically unstable. Dispersing the drug in a polymeric matrix provides enhanced stability to the amorphous form. A typical approach consists in dissolving the drug and a hydrophilic polymer in a common solvent, and after solvent removal, the amorphous solid dispersion is obtained. Here is a schematic representation of a process for the production of amorphous solid dispersions. A common technique used for the solvent removal step is spray drying, in which the solution containing the drug and the polymer is pumped into a nozzle and atomized. The droplets formed are then dried in the drying chamber, separated in the cyclone, and collected. Now, going back to the yeast glucan particles, we then used a similar process for the encapsulation of insoluble drugs. Instead of a solution containing a drug and a polymer, we prepared a suspension of glucan particles in a solution of the drug. This suspension was then spray dried to produce composite microparticles composed of drug and glucan particles. We did an extensive parametric study, but I'm not going to go into the details of it. What's important is that we found out that when we used spray drying conditions that led to the formation of small droplets, the result was a mixture of glucose particles with encapsulated drug and crude drug that precipitated outside of the glucose particles. If the conditions used led to the formation of very large droplets, all of the drug was encapsulated inside of the glucose particles. These images help visualize the effect of droplet size. The image on the right side corresponds to glucose particles with encapsulated curcumin. The sample was produced with the large droplet settings, whereas on the image on the left side, the sample was produced with small droplet settings. In this case, in addition to the glucose particles with encapsulated curcumin, we observe small dots that correspond to pure curcumin that precipitated outside of the glucose particles. The spray drying conditions used also significantly influenced the encapsulation efficiency of the samples. And what's important to point out here is that encapsulation efficiency increases with droplet size. So up to this point, we were able to develop and optimize the encapsulation method by spray drying. But can yeast glucose particles improve drug solubility? The answer is yes. We tested our composites, and not only they exhibit significantly faster dissolution kinetics, but in some cases they even led to supersaturation. And as a bonus, we were able to prove that not only solubility was better, but also dispersion properties improved, as well as flowability. So, to wrap it up, we were able to prove that glucose particles have great potential as drug carriers, not only for macrophage targeted delivery, but also for the amorphization of poorly water-soluble drugs. So if you have any questions, please, you can write me to the email shown here. And also, if you would like to read more about our work, you can find the article published in the link shown here. Thank you for your attention.